I'm just gonna stand there.
Showtime.
There we go. Somebody has to move the slides forward.
Yeah, it's gonna be you because we're up first.
Well, they're getting
Actually,
set up. I'm just going to send around an attendance sheet. We just need names because we need a count for our funders who helped put this all together. So if you wouldn't mind signing in, we promise we're not putting you on another mailing list.
That's the name, I'm trying to say.
Is it like getting recorded so we can download it, or should we be doing
Yeah,
that?
yep. It'll be posted on the Friends of Pika Bay website and YouTube. So,
Send me like a.
I'll send you right. Yeah,
yep,
don't worry
I
about
got you.
Anything to help them get more money.
I will get us started here. So welcome everyone to the 2025 Lakeshore Water Summit. My name is Sam Fromfeld. I work for a nonprofit called Lakeshore Natural Resource Partnership. And my job, we help fiscally manage a bunch of other small nonprofit groups in our area. So we span from the ledge to the lakeshore is our slogan, which is Ozaukee County all the way up to Door County and over to Lake Winnebago. So it's a pretty wide range. And within that range, we have two groups that are really deeply involved in this project, which is the Friends of Pika Bay and Lakeshore Water Institute, which we have representatives from here from Lakeshore Water Institute, Rick and Becky Abler, which are the mentors on this project, who are teachers at UW Green Bay Manitowoc campus. And throughout this process, when I was a student in college, I actually was on the stream team back in the day as well. So it's kind of come full circle that I get to introduce them to you now. So yeah, you guys want to start with your introductions?
For sure.
Hello, and welcome to the 2025 Water Summit presentation. My name is Aurora, and I was the data analyst.
My name is Quentin. I served as the field coordinator.
Hey everyone, my name is Chi and I was the lab coordinator.
My name is Farley, and I was the QA/QC, or Quality Assurance and Quality Control Coordinator.
So as Sam already said, we test various water qualities throughout Manitowoc County. We do this in partnership with LNRP, Lakeshore Natural Resource Partnership. So we are just here to show the data and trends we did throughout the summer.
So our research entailed sampling 25 sites across Little Manitowoc, Pine Creek, and Centerville Creek on Mondays. We also sampled whenever half an inch of rain or more fell within a 24-hour period, regardless of the day, and these were denoted as rain events. Before we start diving into the data, I want to talk about each of the water quality parameters that we measured because each one tells us something unique about stream health. At each site, we measured pH, which tells us how acidic or basic the water is. And we measured this in the lab using a pH probe. Across our sites, the pH ranged from 7.2 to 9.6. And this matters because pH strongly influences chemical reactions and the forms that nutrients and toxins take in the water. For example, at higher pH levels, ammonia increases and certain bacteria like E. coli can survive differently depending on pH. I do also want to mention that a narrow pH range is usually healthier for aquatic organisms, while extreme pH levels can stress out or kill sensitive species. We also measured the turbidity of each sample, which is how cloudy the water is due to suspended particles such as soil or organic matter. We tested this in the lab using a turbidity meter, and this is important because high turbidity can reduce how much sunlight penetrates through the water, affecting how much light aquatic plants receive. It can also clog the gills of fish and often signals runoff or erosion, especially after a heavy rain event or a disturbance. Next, we have our least favorite part, QAQC, which is quality assurance and quality control. This includes calibrating our instruments before using them and running duplicates to check for consistency, while following standard methods to ensure accuracy. Aside from QAQC, a major parameter that we measured was phosphorus. Phosphorus is an essential nutrient for plants, which is why it's widely used in agriculture. However, in large quantities, phosphorus can negatively impact stream quality, causing eutrophication, which is where excessive nutrients cause plants and algae to grow, depleting oxygen levels as they decompose. Over time, this can kill fish and other aquatic organisms, as well as affect the site's long-term health. I should also mention that phosphorus often enters streams from fertilizers, animal waste, and wastewater. So tracking it helps us understand potential pollution sources as well as harmful algae blooms. And then for data purposes, I want to clarify that we measured total phosphorus and total dissolved phosphorus. Total phosphorus is all of the phosphorus in a sample, including the phosphorus adhered to sediments, while total dissolved phosphorus is only the phosphorus that has been dissolved in the water itself. Along with testing phosphorus, we also tested E. coli, which is a bacteria that indicates fecal contamination from humans and animals. I want to emphasize that most strains of E. coli are completely harmless. However, there are some strains that can cause serious illness if ingested, which is why it's a public health concern as well as an environmental concern. To test for E. coli, we used sterilized bottles to collect water samples and then mixed them with a reagent called caloric powder, which is then incubated for 24 hours and then looked at under a black light to see how much E. coli is present. This method for testing E. coli and phosphorus is aligned with statewide methods, and it's the same way that any professional lab would test for E. coli and phosphorus. And then we have ammonia, which can enter our waterways through fertilizers, urine, sewage, and decomposition of organic matter. To measure this, we added reagents that cause color change in our samples, and the color change then reflects how much ammonia is present. Using this data, along with pH and temperature, we can calculate how much ammonium is present. For context, ammonia is significantly more toxic to fish compared to ammonium. And this is because ammonia is more readily absorbed into the bloodstream through the gill membranes, which can cause cell or lethal damage. At higher pH levels and higher temperatures, the proportion of ammonia increases as well. And that's why it's important for us to know what the pH and temperature is, along with the concentration of ammonia in the water. We also measured for dissolved oxygen at each site using a dissolved oxygen meter, which tells us how much oxygen is in the water for aquatic organisms like fish. And then using the same device, we also reported temperature, which is important because temperature can control the amount of oxygen within the water. And then second to last, we measured the flow rates at each site using a flow meter. And this tells us how much water is moving through the stream, which is important because high flow rates can, after a rain event, can flush out pollutants, but also cause erosion and sediment loading. On the other hand, low flow rates can concentrate pollutants, which can then raise the water temperature and reduce habitats for fish. And then finally, the last thing we measured is the depth of the stream using a meter stick to see how deep the streams are, which can change with rainfall. This also influences habitat availability, and together, dissolved oxygen, temperature, flow rates, and the depth gives us the physical and chemical context that we need to interpret our results.
We will now be talking about the 3 different locations of where we sampled at, starting with Little Manitowoc. So for this, you can see that we sampled here in Little Manitowoc near the Lincoln Park Zoo, which is just over there in Manitowoc County. And as you can see, we have codes for each of our 25 sites. For example, LM would be for Little Manitowoc, and then the number of the site. You might notice that it's not necessarily in order because some of the sites were, we got rid of them because they weren't producing valuable data. But you do see that we sample further and further upstream, and these were just in the spots that they were continuously placed. For 2020 to 2021, the Little Manitowoc #1 area was originally done in restoration with LNRP. So now, as you see, with further and further upstream, there are just pictures of each of these sites. The farther to upstream sites, you see a lot more natural areas because they're just surrounded by wildlife and natural plants. But the closer to the city we get with the downstream sites, we are closer to urban areas, as you can see, streets here and here.
A lot of our data that we've tested for phosphorus, ammonia, E. coli, turbidity, has a lot of variables that can confound our data. One such variable is the land and the land use around it and how it affects our data. This is what I was interested in the most. Utilizing ArcGIS, which is a geographical instructional software, along with land use information provided by Manitowoc County, I was able to generate graphs to describe how much and what types of land were around each of our sampling sites. As you can see in this picture here, which is a screenshot directly from ArcGIS, that shows each of the 4 sampling sites for Little Manitowoc, and the color coding tells us what type of land it is. The small dots in the center of these circles are the sampling sites themselves, whereas the larger blue circle represents the 200-meter radius I used to analyze what type of land was in each site. This pie chart here shows the average breakdown for the type of land around the Little Manitowoc Stream. They had a large majority of 51% of natural land, and the second most of which was 13% water. The majority of woodland or natural area might be what contributed to the low average phosphorus slope for Little Manitowoc, which we can see in the graph to the right. On the x-axis, we have our sampling dates throughout the summer. The y-axis, leftmost, represents the average phosphorus in milligrams per liter, and the rightmost y-axis shows us the weekly accumulation of rainfall in inches. The bars along our graph show our total and total dissolved phosphorus. Red being total phosphorus, which is a combination of the phosphorus that's dissolved as well as the phosphorus that is in sedimentary form, whereas the blue bars are just the phosphorus that is in solution. The pink bar along the graph represents the EPA limit for phosphorus in Wisconsin, which is 0.075 milligrams per liter. Little Manitowoc was close, but not quite there at the limit, and it was the lowest among the 3 sites we tested, or the 3 streams we tested on. This is a breakdown of each Little Manitowoc site independently. At the top left, we have LMO1, which is a majority of water at 37%. LMO2 was mostly land used by the Parks and Recreation Department at 35%. LMO3 was again majority woodland at 59%. And LMO5 was a stark majority of woodland or natural land at 95%. LMO5, which had the lowest total phosphorus for any of our sites, along with Little Manitowoc, could be contributed due to the amount of woodland area that it has. In the picture to our right here, we see the stream itself, which is surrounded by natural barrier plants, which can help protect the stream from erosion or sediment entering the water, which would carry dissolved nutrients with it. And the left picture just showing again that woodland or natural area around the site.
Thank you. A big part of this project was monitoring the weather and sampling the sample sites during rain events, which is anytime it rains more than half an inch in 24 hours. So our team wanted to take a look at how this rainfall might be affecting phosphorus levels in the stream and the waterways we tested. To start with, we have, again, the average phosphorus concentration for the entire river in milligrams per liter on the primary y-axis. Then we have the red representing the total phosphorus, which I previously mentioned is all of the phosphorus, including the dissolved phosphorus and any phosphorus adhered to soil particles or any particles suspended in the water. Blue representing just the dissolved phosphorus. Here, we also have on the secondary y-axis, the accumulated rainfall in inches for the week leading up to our normal sampling days, which was every Monday. On the right graph, however, we just have the rainfall that preceded the rain event specifically. Here on the left graph, you can also see the clouds, which signal days that we had rain events, but it also happened to fall on our normal sampling days on Monday. Along the bottom, you can see all of our normal sample dates, and it's all of our rain sample dates as well. You can see, like Harley mentioned, we had quite a few dates throughout the summer for our normal sample dates that were under the DNR's phosphorus threshold of 0.075 milligrams per liter. It's we had 7 days, I believe, that were under the limit. And then you can see where the threshold is as well, indicated by the pink line here. Sorry, and then, oh, sorry. I wanted to also note, with the rainfall, you can see how in the 24 hours preceding the event, it causes, or sorry, after the 24 hours after the rain event, you can see how the phosphorus spikes, but when we go to a few days later, it is mostly normalized.
So while I was watching the 4th of July fireworks, I had a major question of how the fireworks actually affected phosphorus in our streams, because obviously we produce a lot of fireworks on the 4th of July, and that sediment gets washed down into our streams. So I did some research, and on average, the United States produces about 420 million pounds of fireworks every year, which is a lot. And in 1906, at the Berne Convention, in the United States, it was banned for white and yellow phosphorus, which was used previously as dye for the fireworks. But red phosphorus is still allowed, and every little bit adds up with the amount of fireworks that go off every year. And again, as Chi stated earlier, phosphorus creates eutrophication, which is excess nutrients, which sucks the oxygen out because it decomposes from the algae, and then it will degrade the water quality over time. So what I did was that I looked at the sample dates for all of our sites, starting with Little Manitowoc. And again, you can see that for the most part, the sediment is lower than the standard for the EPA, for the most part, except right after the 4th of July, when we did sample, as you can see with the star here, it is significantly higher than the rest of the entire summer. And that's with other factors, including spreading manure or phosphorus fertilizers out on lawns, just the heat and temperature. Many factors will affect the amount of phosphorus, but you can see a stark contrast with the amount of phosphorus. And most of it, you see a big jump between the total dissolved, which is just diluted into the water, versus total phosphorus, which is phosphorus in sediment. So you can see a lot of it is sedimentary, likely from the fireworks. So there's a big jump that is almost double the amount. And then overall, you see that there's a jump right back down the week after with the amount of rainfall. So you can see that there is a noticeable difference just from the fireworks alone in Little Manitowoc. So for Pine Creek, which is our next location, we will show the pictures that are in Newton. Newton, all the way over here, as I'm sure you are mostly aware of, is a lot farther south of Manitowoc. And we have 8 different sites, which is double the amount in Little Manitowoc. Again, they're not necessarily in order. We have PIO2 over here, which was the second one that was put in, and then we have PIO6 and PIO5 all the way up at Karstens Lake. PIO6 runs into PIO5, and then it keeps going down into Lake Michigan. PIO8 is just a little area in a farm. It is not necessarily a part of this because, as you can see, it is broken up. So for the northern 4 sites, you can see they are mostly just surrounded by either farms like the PIO7 or just natural areas. You can see there's a bit of algae growth in PIO6 where it leads into Karstens Lake, and it comes out into PIO5, which has a lot of murkiness, probably a bit of movement from the person sampling in there when I took that picture, and a lot of wildlife just in general for all of these sites. And then same for PIO8, PIO3, all of the southern sites. You can see a lot more of just natural area compared to Little Manitowoc. You see an abandoned railway where we sample at PIO3. A lot of these are actually near farms, whether it's across the road or just directly next to it, like for PIO2. Overall, we didn't have any restoration areas, but the county itself does a little bit on smaller scale, just like to maintain the sites in Newton or Pine Creek.
Pine Creek, which had the most agricultural land by far compared to all our 3 streams, also had the second highest phosphorus. We can see in this ArcGIS screenshot depicted by the orange, the amount of agricultural land that is around our sites. This pie chart here shows the breakdown by percentage for the Pine Creek land use, with the majority being 48% agriculture and the second most being 19% natural or woodland land. Among the Pine Creek sites, I noticed that those with higher agricultural percentages also had a higher difference between total phosphorus and total dissolved phosphorus. The sites that had the highest difference were PIO5 and PIO6. PIO5, which is located near Sonnenberg Lane, and is the second most upstream site, had a 62% majority of agricultural land. We can see in the photo here, the farm that is near the stream. And then the right two photos, we see the stream itself, having some barrier crops, but still a little bit of the stream being affected by runoff and erosion. PIO6, which was northeast of PIO5 in Carson's Lake, had one of the highest percentages of agriculture at 85%. We can see the stream is right here in between these two, and it was actually located near another LNRP managed area. The second most percentage for PIO6 was the road, as depicted in the picture by the bottom right. These two graphs show the phosphorus for PIO5 and PIO6.
Wow.
Along the x-axis, we have our normally sampling dates, and the y-axis shows the phosphorus in milligrams per liter. Our blue bars are representing the total dissolved phosphorus, or the phosphorus that is in solution within the water. And the red bars show the phosphorus that is both in solution and out of solution or attached to sediment. We can see large gaps between the blue and red bars, indicating the variance between the phosphorus that is dissolved and the phosphorus that is non-dissolved. This graph here was created to show the correlation between agricultural land and the amount of phosphorus that was not dissolved in solution. On the x-axis, we have the agricultural percentage for the land use, and the y-axis shows the percentage of non-dissolved phosphorus within the stream. We can see a positive trend line here showing that there is a correlation between the two. Now, for those of you that don't know, an R-squared value is a measure of statistical certainty, describing the variance between data points and the distance from the trend line. With an R-squared value of 0.3679, there is a moderate to weak correlation between agricultural land and the difference between total phosphorus and total dissolved phosphorus. I also thought that there might be a correlation between the agricultural land and the levels of E. coli that we were testing for. This graph here was created to illustrate. The x-axis shows again the agricultural percentage around the stream. The y-axis here shows the E. coli levels measured in MPN per 100 milliliters. MPN, which stands for the most probable number, is how we measured E. coli over the summer. This is different from another industry standard, CFU, or colony forming units. MPN, which is what we used, is a statistical estimate of the amount of E. coli, as opposed to a direct count of colonies like CFU. As we can see with an R-squared of 0.0161, we cannot say that there's a statistical correlation between agricultural land and E. coli levels in Pine Creek.
Again, these graphs were created using the averages from all of the sites on Pine Creek to get the totals. We still have the average phosphorus in milligrams per liter on the primary y-axis here. And then we have, again, the red representing the total phosphorus, which is the phosphorus dissolved in the water, in addition to the phosphorus adhered to sediment in the water. Then, represented by the light blue line, again, we have our rainfall in inches, again with the left graph showing the weekly accumulated rainfall prior to the sampling date, and the right graph showing the rainfall for the 24 hours leading up to our sampling date for the rainfall events, which is anytime it rains more than half an inch in 24 hours. The x-axis, the clouds are still above the rain events that we had fall on our normal sample days. The x-axis still shows all of the dates that we went sampling as well. Something to take note of though is that we didn't have a single sampling event where the average for the creek was under the DNR's threshold of 0.075 milligrams per liter. The lowest we had was approximately 0.829, that's a rounded number. And then we also saw a correlation between rainfall here and an increase in phosphorus, as well as we see that over here with our rain data as well. something to take note of. There is a spike in rainfall here, but we don't necessarily see that, or sorry, there's a spike in, sorry, there's a spike in rainfall here, but we don't necessarily see that come through in the normal sample dates, but we see it more in the rainfall events. This is likely because most of the effects of rainfall are washed away, for lack of a better word, within 24 hours of said rainfall event, but we still see how the rainfall in the week leading up might have an effect on the total phosphorus or all phosphorus levels. Yeah, and
So for the firework effect on Pine Creek or Newton, you can see again, it gradually increases in phosphorus in general throughout the summer. But right when you get to after the 4th of July, it peaks right at the top and then it goes right back down in phosphorus in general. But as the summer continues towards the end, there was a lot more rain as well. There seemed to be a lot more phosphorus in the end. It could all again be from more manure or fertilizers being spread, the heat, a lot of factors can play. But again, there's also, it's a lot of washing from the rain. You can see a slight trend in the amount of rainfall, as Quentin already pointed out. You see it increase with the amount of phosphorus. But I would also just like to point out that my rainfall is actually an average amount throughout the week. So I took his cumulative and then I divided by 7. So you can basically see an average per day instead of the cumulative throughout the week. But you do see that there is still a significant amount, just to notice that there is more sediment from the fireworks after the 4th of July than the rest of the surrounding time frame, and then it just goes back up, but that is not necessarily from the fireworks.
Yeah.
So lastly, for Centerville, this is our 3rd location. This was in Cleveland, so now we will talk about the data for this. So Cleveland, even farther south, long drive for me, to be honest. You see 13 different sites. That is more than LM and Pine combined. That is, I mean, one more than, but still, that's a lot of work for me, to be honest. So you can see that there's 2 branches of the stream actually that go into Lake Michigan. One goes through Lakeshore College, 2 of them actually, CN09 and CN08. The CN is Centerville North, CS Centerville South, and then CR Centerville Restoration. So for the Centerville North sites, we have 5 of them. Again, you see a lot of just surrounded by natural areas. Some are with farms. You can see a farm right behind CN10 and right across the road from CN11. And then CN08 and CN09 are through Lakeshore College. And then CN07 is kind of just in the middle of nowhere. And then you got the south sides. These technically do look split up, but they do actually connect, even though the map didn't like to show that. CS13, this is near a farm. CS14 is both near a farm and also just like kind of in the middle of a road somewhere. CS06 is next to a park. And then again, you just got kind of middle of nowhere places for these 2. And then the Centerville Restoration part, this was called CR because it was restored in 2012 from LNRP. So you can see that like it just goes right past Hyka Park into Lake Michigan. And you see that it has a lot of open bodied water and it's surrounded by a lot of just natural areas. And you can see the difference. This was before the restoration where it's very flooded. And then after they did the work, look how great that looks. That was phenomenal. You can actually see that it's a stream now. This is surrounded by urban setting, even though it has a bunch of natural area around it.
As we can see in the photo from ArcGIS here, Centerville had a much more diverse distribution of land use compared to Pine Creek. The pie chart is as follows, with 29% being agricultural land and a majority at 43% being natural or woodland land, as Aurora said. Now, Centerville had the highest average phosphorus by far compared to the other streams that we tested. We can see again in our x-axis the sampling dates that occurred over the summer. In our leftmost y-axis, the average phosphorus in milligrams per liter, and our right y-axis showing the cumulative weekly rainfall leading up to our sampling events. Our red bars indicate the total phosphorus. The blue bars represent just the dissolved phosphorus for what is in solution. We can see that none of the sampling dates are even close to being at that EPA threshold of 0.075 milligrams per liter. Like with Pine Creek, I noticed a correlation between a higher agricultural percentage and a higher difference between the total phosphorus and total dissolved phosphorus. In our x-axis, we have the agricultural percentage of the land around our sampling sites, and in the y-axis, we have the percentage of non-dissolved phosphorus. Again, the trend line here shows a positive slope, indicating that there is a correlation, and our R-squared value is even higher than that of Pine Creek's at 0.4433. Now, part of the larger average phosphorus in Centerville was due to specific sites. One such site is CS13 with an agricultural percentage of 81. We can see in the picture to the right here, this is the farm that was adjacent to CS13, roughly 50 to 100 meters away from the stream. And in the leftmost picture, we have the stream itself. You can tell that the width, especially compared to the Centerville restoration, it's much lower. And there is no natural barrier around the stream itself, making it very susceptible to erosion and sediment leaking into the stream. Another such site was CN10, which had one of the largest agricultural majorities by far at 74%. Now, other than high phosphorus, I noticed that these sites also had higher E. coli, which led me to believe that there might be some sort of correlation between the agricultural land and the E. coli levels. As we can see here, again with agricultural percentage on the x-axis and E. coli levels in MPN per 100 milliliters on the y-axis, there is a somewhat positive slope on our trend line, but an R-squared value of 0.108 is still too weak to say that there's a statistical correlation between the two.
Thank you. Again, these graphs were made using the averages from all of the sites on the creek. We have the phosphorus in milligrams per liter here on the primary y-axis, red representing total phosphorus, blue representing just the dissolved phosphorus. The light blue line indicates rainfall. On the left graph, it indicates the weekly accumulated rainfall or all the rainfall that happened in the week leading up to our normal sampling date, which was every Monday. Then we have the rainfall on the right graph showing just the rain that occurred in the 24 hours preceding the rain event, which is whenever it rained more than half an inch in 24 hours. Along the x-axis here, you can also see all of our normal sample dates and all of our rain sample dates. The pink line still indicates the DNR phosphorus threshold. Something to note with this, with these levels though, is this was heavily skewed by the hotspots we had at CS 13 and 14. The averages were much higher with them included. Phosphorus pollution still remains like a very significant issue in Centerville. As you can see, it had, like Harley said, the highest phosphorus levels on average out of all of the waterways tested. And you can also see how it still appears to mostly follow the amount of rain in the week leading up, and we see the largest spike here too. Next slide. Little Manitowoc had the lowest levels of nutrients on average, but we still see higher phosphorus and ammonia after rain events. The low averages recorded in the Little Manitowoc River suggests that the surrounding vegetation and natural areas are reducing the amount of nutrients that are being washed into the river. Pine Creek did have higher averages than Little Manitowoc did, but we also saw how sometimes, even with a lot of rain, it doesn't always create a spike in the week or in the normal sample day. In the normal sample dates, we saw that the increase in rainfall didn't necessarily increase the phosphorus level, but we did see that the rainfall increased it in the 24 hours after. Centerville Creek also had the highest phosphorus tested, as previously mentioned, with one event having nearly 10 times the DNR's phosphorus standard for total phosphorus. We observed that rainfall increased phosphorus levels, tended to increase phosphorus levels with the in the rainfall in the week leading up to our normal sampling dates tended to have an effect on phosphorus levels. This correlation could be due to increased decomposition of organic matter in the soil, facilitated by the water necessary for decomposition. Also, I guess I already mentioned that, but ultimately, most of the effects that we see from rainfall are in the 24 hours after a storm, which is when we'll see most of the nutrients from the surface flow and subsurface flow entering the river.
Finally, for the Cleveland firework effect, you can see, once again, there is a steady increase throughout the summer when we sampled, and then a spike after the 4th of July. And you can see a big jump in between the dissolved phosphorus, which is just in the water, and then the total phosphorus, which is the sedimentary phosphorus. And then it goes back down. It immediately goes back up because it's Cleveland. They have a lot of phosphorus, as you can tell, compared to Little Manitowoc. So technically, the amount of rain and like phosphorus, it's not going to do too much to it's not going to get rid of it as well as we would like for it to go, but it goes back down again after that big spike in the rain, and then it goes back up throughout the spike in rain towards the end when we had a lot of rain events. So overall, we can see just in this area for the sample events, there is a big spike after the 4th of July. And I don't know about you, but I haven't seen a whole lot of fireworks in Cleveland. So this is likely from Manitowoc when all the fireworks go off after the 4th of July. It is sediment in air, so it gets blown throughout the night and then it lands in places like Cleveland. And then it gets washed away with the amount of rain because we had a rain event right before it. So you can see that there is just going to be a lot of it because it's going to move in the air.
Cool. I'm ready up next, it seems. Next, we want to talk about hotspots, specifically site CS13 and how agriculture, rainfall, and runoff are impacting water quality in this stream. My goal for this section is to highlight why CS13 is considered a hotspot and what makes it different from other sites in the watershed. But towards the end of this section, I'm also going to be talking about PI05 and comparing that to CS13. So before I begin, CS13 is the most upstream site located at South Union Road on the Centerville South Branch. What makes this site important is its land use, with over 81% of its land being agricultural. This means that field runoff regularly enters the stream during rain events, carrying nutrients and sediment. After storms, the water often turns muddy and we see clear spikes in phosphorus and ammonia levels. This tells us that CS13 is a hotspot directly influenced by agricultural runoff. Going more into land usage, this slide shows a breakdown of land use around CS13, with more than half the watershed dominated by agriculture. Because of this, stormwater carries fertilizer, manure, and soil directly into the stream. To provide a point of comparison, I've also included a pie chart of CS14, which you guys saw earlier, and which is the next monitoring site downstream of CS13. This allows us to contrast the land use patterns between CS13 and CS14 and begin to see how differences in land usage across sites may influence water quality throughout the watershed. Aurora's favorite photo. This photo on the slide was taken just outside of CS13, and to the left is the direction of the stream itself, and on the right you can see the surrounding area, which is entirely farmland. I also want to mention that the slope from the farm field to the stream is significantly steep. During rain events, fertilizers, manure, and other materials from the crop fields wash directly into the stream. These runoff events are strongly tied to the elevated phosphorus levels and other nutrient inputs, which can degrade water quality that we see at CS13. This photo is an example of what runoff looks like after a rain event. If you look closely, you can see the streaks and marks left behind by the runoff. All of that rain carrying fertilizers and nutrients from the fields can flow directly into streams, which is why rain events often transport pollutants from land into waterways. So going back to CS13, let's look at the data and see how rainfall affects this site compared to other sites. The top left graph shows South Centerville during dry conditions, while the bottom left graph shows data during rain events. Phosphorus is measured in milligrams per liter on the left y-axis, and turbidity is measured in NTU on the right y-axis, with the different sites on the x-axis. The takeaway from these two graphs is that compared to other South Centerville sites, CS13 consistently has the highest phosphorus levels, and we see those levels rise even higher during rain events. If we look at the graphs to the right, which displays ammonium and ammonium levels in milligrams per liter on the y-axis, and the sites on the x-axis, we see the same pattern as phosphorus levels. CS13 towers above other sites, both under dry conditions and after rain events, which is why we identified this site as a hotspot. I want to also clarify again that CS13 is our most upstream site in the South Centerville branch. And this is important because this site affects all of the other sites downstream of it. For example, we see that CS14 is high in phosphorus levels. However, we can hypothesize that some of this phosphorus is coming from CS13. That is really site action. Going back to this photo of CS13, we noticed a strong manure and sulfur smell at the site, which is most likely contributed from the manure spread on the farm. The first thing you'll probably notice as well is the heavy algae growth along the stream, which makes sense given the high phosphorus and ammonia levels that we saw in the data. These conditions create the perfect environment for algae to thrive, creating algae blooms and affecting the stream's health over time. Going to PIO5, so PIO5 is a site in Pine Creek, and we're just going to be comparing to CS13. These graphs shown on the screen display the phosphorus levels for sites PIO5, which is one of our lower phosphorus sites within all the streams. So then phosphorus levels for sites PIO5 and CS13 is displayed in milligrams per liter on the left y-axis, and turbidity and NTU on the right y-axis, with the sites displayed on the x-axis during dry conditions and rain events. Right away, we can see that site PIO5 has much lower phosphorus levels compared to CS13. And the reason for this is because of the different land management practices that each site implements. This photo is from PIO5, which serves as our lower phosphorus reference site. And right away, we noticed that the water had far less algae and was clearer during rain events compared to CS13. It also lacks the strong smell of manure or sulfur, and these differences highlighted how significantly agricultural runoff influenced conditions at CS13 compared to PIO5. What we found to be interesting is that when we looked more into PIO5, we noticed that PIO5's land usage is heavily agricultural as well. However, it still had significantly less phosphorus during both dry conditions and rain events, with less turbidity as well. And the reason for this is because PIO5, a site in Pine Creek, implements cover cropping. For those of you who don't know what cover cropping is, it's when specific crops like cereal or rye are planted to provide continuous cover for the soil between cash crops. So for instance, in this photo, you can see all of the dead white grass with the corn growing between them. It's kind of a blurred photo, but this white grass is cereal rye that has been terminated, which is used as a cover crop. And this has significant benefits such as reducing sediment runoff and nutrients runoff. This is because the dead rye holds the soil together and also reduces nutrients from washing away during rain events. And then using photos from the Bodas family farm, I think these photos do a better job at visually explaining what cover cropping looks like in practice. On the left photo, you can see cereal rye that's been planted as a cover crop. This rye is then terminated, usually by spraying before the main crop is planted. In the middle photo, you can see the newly planted corn growing directly between the terminated rye. Like I mentioned earlier, one of the main reasons why we see such a significant difference in sediments and nutrients runoff between PIO5 and CS13 is due to cover cropping. Cover crops like cereal rye are incredibly effective for several reasons. They reduce sediment and nutrients runoff, which protects nearby waterways, and they also suppress weeds because the dense mulch from high carbon rye creates competition and smothers potential weed growth. Not only that, but they also contribute nutrients back to the soil as the rye decomposes. And then you guys saw this graph earlier, but just coming back to this graph, when we compare data from rain events to non-rain events, we can tell that cover cropping is significantly reducing the amount of phosphorus and sediment loading into the stream. This is why during rain events, the phosphorus and turbidity levels, this is why during rain events, the phosphorus and turbidity levels decrease at PiO5 while CS13 increases because the runoff is carrying fertilizers and sediments into the stream. So basically, the water is diluting it, right? I don't know. To wrap up this section, what you basically need to know about CS13 is that it's the most upstream site in the South Centerville branch, where rainfall and surrounding land use strongly influence the site's water quality. The data shows that phosphorus and ammonia levels increase significantly after rain events, and in the field, we see the effects of this, such as algae growth and cloudy water. When compared to lower phosphorus sites like PiO5, that difference becomes even more apparent where PiO5 doesn't show these same effects. The key takeaway from this section is that rainfall and land use together plays a major role in shaping stream conditions, and CS13 gives us a clear example of how watershed characteristics influence water quality and why it's important to continue monitoring and learning from these sites. Sorry, I said PiO5 doesn't show any algae. It does, but it's just not as high as CS13. So yeah.
One of our wonderful partners for this project was LNRP, or the Lakeshore Natural Resource Partnership. And they participated in a restoration project where Centerville Creek, near where Centerville Creek empties into Lake Michigan. Among other things, they sought to mitigate the erosion of sediment that had accumulated from when a dam was built on the creek in the 1890s. And the dam was eventually removed in 1996, while initial restoration work started in 2012. So our team decided to examine how effectively the restoration area is performing its job reducing erosion. The restoration area surrounds CR01, CR02, and CR03, just west of Hika Park here. You can see here the area that the reservoir used to cover before the dam was removed. Additionally, you can see where the north and south branch, the pointer doesn't really work, but you can see where the north and south branch meet on the bottom image there. A variety of methods were used to help control erosion, including tree planting, physical bank protection, and incorporate a comprehensive redesign of the river, which incorporated bends and ripples to regulate water speed. With these graphs here, we have the rain events on the right side and the non-rain events on the left side. Again, the phosphorus is in milligrams per liter on the primary y-axis. This time, however, on the secondary y-axis, it is turbidity, which is measured in NTU. Then highlighted here, or I guess red still represents total phosphorus, which is the phosphorus dissolved in the water as in addition to the phosphorus adhered to particles in the water. The blue represents just the total dissolved phosphorus. And then again, the green line represents the turbidity of these sites. The brown tannish box here is meant to signify the 3 sites that are a part of our restoration area, or the LNRP's restoration area. And the pink line again represents the DNR threshold for phosphorus. We looked at how it was doing at reducing erosion by looking at the phosphorus for both the rain and non-rain events. We do see higher averages. We do see higher averages for the, compared to part of the south branch, in the restoration area. But overall, the turbidity doesn't change that greatly during non-rain events. However, when we look at rain events, we can see that the turbidity massively increases inside the restoration area. And in some cases, or from CRO2 to CRO1, the turbidity actually increases inside the restoration area. Something to note, though, is how the phosphorus remains mostly stable, even with a massive increase in turbidity. This kind of suggests that the restoration area is doing part of its job in removing nutrients from the surrounding area, preventing them from being washed into the lake or creek and then the lake. Over the summer, we did observe significant erosion inside the restoration area while we were sampling. Primarily in the form of riverbank and gully erosion. The photo on the left shows a picture of a riverbank near CRO2, where the dirt underneath all of the grass has been washed out, and clumps of sod are kind of falling into the river as they get the dirt from underneath and gets washed out. And the two pictures on the right show a picture of gully erosion. It may be hard to spot between all the grass, but there is a trench going through right there. And the next one. Here, you can see we have graphs representing flow rate for Centerville, both for rain events and for non-rain events. Again, tan box highlighting all the restoration area, and the stream and the y-axis showing the stream flow in feet per second. So you can see how no matter whether it's rain or non-rain events, the water speed peaks in CRO3 during non-rain events, and then CRO2 during rain events. CRO3, just to note, is where near where the restoration area begins on the upstream side of the creek. We also see the flow rate decrease during non-rain events, which is a good thing, but we see it increase at CR2 until CRO2 during rain events. Next slide. Oh, I forgot to mention, sorry, but with the increased flow rate, we do expect to see more erosion. I should have that as a note here, so.
So as with a lot of the data we were testing for, as we said prior, there are a lot of confounding variables that may make analysis for the data difficult. One such confounding variable that we are able to analyze is the land and the land use around our sites. We've seen the correlation between agricultural land and phosphorus levels, particularly sedimentary phosphorus or phosphorus that is not dissolved within the stream. While agriculture is important, it's a backbone of our country, so is preserving our water. We have to take agricultural practices and land management practices into consideration when asking ourselves how we can preserve our streams and prevent further nutrient and degradation to our streams. As Chi mentioned with PIO5, activities such as cover cropping can help to minimize the amount of nutrients that get washed into our streams or the amount of erosion that ends up degrading our streams. The streams and creeks we tested are largely fed directly or indirectly by rainwater. Dissolved nutrients such as phosphorus can be washed into the stream by rain, which also causes erosion and leads to more sediment and bank erosion. Now, the restoration area, as Quentin just talked about, should be monitored and continue to be protected to preserve our environment. It is a such example as how our efforts and engineering can protect our streams, but they're not forever. It's a temporary fix, and they need to continue to be monitored. While we cannot say for certain that there's a significant effect from fireworks impacting phosphorus levels, there was a noticeable increase after the 4th of July with the levels of non-dissolved phosphorus that we have seen. And it suggests increase from runoff from fireworks sediment. Hotspots are areas that show consistently higher levels of nutrients, especially compared to other sites. They often respond strongly to rainfall, causing spikes in phosphorus, ammonia, coli, and turbidity. They highlight where water quality is the most vulnerable and where management efforts can have the biggest impact.
Any questions?
Yeah. Yay.
Yes.
What types of plants are used in cover crops? You mentioned one,
Oh,
and I'm a little hard of hearing, so
I see.
it's okay.
So the specific cover crop that was used in the site that I mentioned was cereal rye.
Right,
I said it, so technically any crop can really be a cover crop,
right,
but most of the time it's used as, most non-cash crops are used as cover crops. So cereal rye within Wisconsin that I'm aware of is not typically a cash crop like corn.
right.
I'm not
I
100%
know a little bit
sure.
about cover crop stuff.
Yeah.
So like rye is really helpful because it has a fibrous root network, a lot of below ground organic matter, and all of the above ground stuff. Basically, it's really good at holding the soil in place. Sometimes though, farmers will mix in multiple different plants with their cover crop spread. Oftentimes they'll put in legumes or other plants like that that are able to fix nitrogen from the atmosphere and add fertility to the soil.
Right.
And so it really depends on each farmer's specific needs that they're gonna like tailor that cover crop to their land kind of thing. You know what I mean? Very helpful tool, but
Sounds like something that deserves a little more development.
oh yeah, there's a lot of research. We didn't cover a lot of that stuff because that's not the focus of our research.
Sure.
But there's a lot of research that has gone into cover cropping, especially at like UW-Madison, I think, and stuff like that. There's been a lot of research, but
It sounds like a cost-effective tool. It's,
Yeah.
it's,
the
that is in the end what it all comes down to,
Yeah.
is what we can afford to do.
Exactly. There are some downsides, of course, like she mentioned, they had to spray to terminate the rye, which is typically not ideal because of the herbicide runoff that can.
Collateral damage.
Yeah, exactly. And so there are other methods, but they're harder to do at mass scale. Sometimes though, farmers will plant crops in the fall that die over the winter, so they don't have to kill them off in the spring.
Sure.
But again, it all heavily depends on.
Sounds like a good area to develop in terms of,
Yeah.
you know,
Protection, like, you
you're
know,
looking at a GMO type thing.
Yeah.
You know, you could do that in the cover crop field.
or even like GMO cover crops, make them more better at their jobs. Like it's a very interesting thing,
A
yeah.
cover crop that could, after it dies off, partially replace phosphorus and fertilization.
Yeah,
And-
exactly.
and Reduce the amount needed.
And
Yeah.
GMO cash crops help protect it from the spray such as Roundup that's used to kill the cover crops.
Well, yeah, that's the thing. So like, yeah, that's what, yeah, you said, sorry, got confused me for a second there, but yeah. He just said that, basically a lot of cash crop, or not a lot, but cash crops that are used with spray, cover crops are often genetically modified to be, like, I think the term was, I've heard commonly like Roundup ready, and stuff like that, where they're,
Resistant to,
yeah, like glyphosate and other herbicides and stuff.
resilient against, I should say.
Exactly, and so like, while the cash crops may be fine, the non-resistant, cover crops would die off.
Right.
Yeah.
Sounds like a good avenue to pursue.
It is a very interesting field of study, I will not lie, I like that kind of stuff. Any other questions?
So the research that you've done is great. Tell me what next steps are. Where does this go from here? And what is it going to benefit?
Well, from here, I believe the plan is to still continue to monitor, the restoration, at least Centerville, but, the sites that are actually selected each year, change. No, not too much?
Not too much.
Not too much. I remember we got rid of,
We
Oh,
have
okay.
looked at some, they're pretty stable
Okay.
now, but, I guess we can speak to some of that
Yeah.
too, just because we do have to have the multi-year view. So obviously, they've collected a lot of data this year, and as they said, at least on Centerville, that data collection goes back pre-restoration to 2010. So, yeah, so what are we doing with all of this data? The goal is to basically help land managers and conservation groups use this data to understand where the best places are to put in certain management practices such as cover cropping, harvestable buffers, major restoration projects like LNRP is doing. And so this is where we work with our community partners like LNRP and Manitowoc Soil and Water Conservation. So actually the cover cropping that she talked about is part of a larger initiative through the county where they're looking at different places along Pine Creek and saying, all right, which of these smaller, more cost-effective maybe interventions really are feasible for these farmers and land managers to implement? So things like buffers, things like grass waterways, things like covered crops. Whereas we're working with LNRP right now to assess, we see some erosion happening and some things degrading this awesome restoration project they did. Where can we target that? So that's really the goal of what the students do. And then our job is to work with those partners to continue maintaining these restorations.
And then beyond our smaller project, the data the students are generating is also being used in larger scale management practices. So Pine Creek is part of a 9 key element plan, which was developed partly in response to the higher phosphorus and turbidity loading in that creek. And a lot of the watersheds that we're looking at are part of what's called the TMDL, total maximum daily load, where sources of phosphorus are assessed, load limits are set, and that the goal is to ultimately bring the phosphorus levels down below that threshold. So there's a lot of projects like Becky said, from the small, easy, relatively inexpensive projects to these larger watershed scale projects.
And if you didn't see it in the gymnasium, there was also a project this summer near one of our sampling sites. It was using an experimental phosphorus removal system using activated alumina. We saw that right next to one of our sampling locations. The colloquial term for it is Poseidon pellets. And experimental projects like that help to reduce the amount of phosphorus in our streams. And while it's not the most cost effective, especially compared to something like cover cropping, it's still something that, again, could become cheaper in the future and is worth looking at for land managers and farmers.
Yeah.
I have two questions. The first is, you're the youth for our future. So what was the most exciting, most revelation idea that you got out of your project? What really was an earth shattering, like, oh, we've really got to work on this for future versus like, oh yeah, okay,
What were
we
the
all knew that kind of thing.
any epiphanies?
I was shocked by the difference in phosphorus levels between Little Manitowoc and Centerville. Like the large gap there, even with all the restoration and stuff that has happened in Centerville, it to me stands out like that is like definitely an area of interest. So like, why is it different? How can we make Centerville more like Little Manitowoc in that sense and lower those levels? But
For me, I'm really big on like just land restoration in general. So like planting trees to create barriers for erosion, doing like the logs to help mitigate the barriers from cutting across the bank, stuff like that. That's something that I'm really passionate about and I'm hoping to do in my future career.
Something for me that stuck out was more on the methodology side. For our phosphorus testing, we did it like the old fashioned way. So we would have all our beakers out and then we would manually add our reagents such as acid, put it in an incubator of sorts, which just to heat it up to really remove any organic matter that was in there. And then, we manually tested it using a spectrophotometer.
I messed up a lot.
No. We didn't mess up at all.
Another project that she was working on, they had a machine that actually did it all for them in one foul swoop. And so I thought that was pretty neat, just the advances that are being made in the analytical side.
Yeah. I wish we had that.
So if I won the lottery tonight and wants to buy that.
Yeah. Like you say,
I'm sure they'd be happy with
so
that.
many hours of like work that we spent just because you have to filter some, like half the sample, you have to make filtered samples and unfiltered samples, then you have to stabilize them with acid, and then it's just a whole process with all the reagents and everything. It took a while, but.
Some of our lab days, we were doing 120 samples.
And then messed up 50 of them.
120 or is it
Yeah.
100 and, was it 107, I thought? Yeah.
Along with
or-
our QAQC, and then if we messed up.
If we messed
150
up,
with the
yeah, I guess.
mess up, yeah.
Okay, maybe, but like,
They worked hard.
Um,
Let's leave it at that.
So in my current projects now, I don't need to deal with any of that. So basically, the machine that Harley's talking about is the AQ300, which I use for my current project right now. So the epiphany that I had while doing this project was actually like, so we're finding like problems with a lot of these sites, like the phosphorus, right? But then my big issue with it was that like, oh, I'm discovering all these problems, but like I'm not doing anything about it, right? I mean, I am, but like, I don't know.
yeah.
So, but basically what I did then is, together with, Michael Holly, who's a professor at the Green Bay campus, Beth Condro and Andrew Bodas, we're basically working on these, phosphorus prototypes, it's a project that, some, like the high schoolers were kind of working on a little bit. But basically the goal of these phosphorus prototypes is to, we're eventually gonna implement them. Well, so for now, we're gonna implement them in smaller scale stuff until we know if they work or not. But, so talking about the activated alumina, actually, that Harley was talking about, we kind of discovered that this product, it doesn't remove the amounts of phosphorus yield that we would want from, the like higher phosphorus like streams or stuff, right? So we're essentially we're trying to create a product which we're calling it aluminum hydroxide for now, which it does exist on the market already, but we're trying to create a product that would remove more phosphorus, right? And so far, actually, with some progress, right? And this is thanks to Beth mostly, but like we've actually been able to like with this product, like we've discovered that it removes a higher amount of phosphorus than most other products that we could find in the market, right?
Through a chemical or through a chemical combining, compounding, or how does it remove?
It is absorption.
Yeah, this question is honestly such a better question for Beth, since she's working on it, but I just know kind of like the idea of it basically. I believe the goal with it is to basically, I would assume that the phosphorus is being adhered to the product, right?
Oh, bonded.
Yeah.
It makes sense.
It's a physical removal. The electrons in the phosphorus, the
Right.
electrons in aluminum or aluminum hydroxide
Okay.
absorb. So it's just the electronegativity pulls it to it.
Yep.
And so it's not forming a new compound by any means, but.
Right. So
Yes.
the physics of it, if you will. And the electric.
Yeah, and I guess if any of you guys have, like, if you want to see the project, I do have photos of it. So if you want to like look at it afterwards, I'd be more than happy to show you.
Interesting.
Yeah.
Somebody have a question?
Yes.
Oh, yeah, Ash.
Did you have any fun or funny moments during the whole thing?
I do,
Oh.
I do.
Oh, no.
Oh, no, I actually have a video and I can show you guys it later. So I got these really crappy boots because I didn't get in the water the whole summer. I didn't fit in the waders. So my boots had holes in them, right? And the stream, like the erosion site where, it was like the ground was breaking apart,
No.
that Quentin showed, that was the trail that I was walking on. And it was very muddy and rainy, so it was not good conditions to walk back. So Harley ended up carrying me the whole way through the site on his back. I have a whole video of it. It was amazing. And then the high schooler behind us fell in the water carrying the stuff.
Yeah.
So,
Things we do for science. right?
yeah, things to do.
The high schooler in question later fainted.
Oh, yes, that was.
That was a different day.
That
That was a
Biology
different
and physics. got it.
Yeah. Gotta drink water when you go out to the field.
Yeah.
Heat exhaustion is a real bad thing.
We have also had days where a lot of us were gone because it is 4 of us and then the 2 high schoolers. So some days we actually had people in our lives come sampling with us. So my fiance, my friend, and my mother actually joined us for, I think, 2 or 3 of those sampling events just to help us out. So that was a lot of fun too.
I slid down one of those, yeah.
She did fall.
I think you get some credit somewhere, right?
No, technically,
They're
yeah.
all in the credits there.
The family, in your last name.
Oh, yeah.
Yes.
I just think I also heard of an evening where two of the boys went out, men went out, and it was after rain, and there were many mosquitoes.
These two, right?
Yeah, that one was a lot of fun.
The mosquito repellent did very little.
Yeah.
They don't.
I brought it for them because they always forgot and it did nothing.
Yeah.
Going for dinner.
It did not deter them.
Yeah, no, it made them hungrier, to be honest. Yeah.
Yes.
Some seasoning to the meal,
So,
add some seasoning to the meal.
Yeah.
yes, yeah, it made it flavorful.
We have a question in the back,
Yeah.
right?
Couple of questions, let's see.
Yep.
You know, the lunch tray is a lot more useful if you have a lighter.
Ah.
Yeah,
I was just, it's like, did a light.
yeah, like in the Bee Movie.
Oh, gosh. All right, other questions? Oh,
Are you gonna, I mean, from this point on, are you gonna get a job? Are you gonna develop? Are you gonna?
Oh, me, her,
Hopefully a job.
I'd like to have a
Like
job.
that.
You know, personally, I'd like to be employed.
Yeah, if anyone's hiring.
Consulting firms.
Yes, of course. Yeah.
All right, well, thank you.
Thank you. Yeah, thank you. Good, guys. We're finally done. No more presentation to look forward to.
Graduation cap.